very much impressed that okay, it's obvious that the mutated version will be at the lower energy and further. That's why further exertions of this virus we can predict that it will be pandemic or any infections. So now flu, that many times now we are having flu, having the similar kind of virus you can get, but the mutated version, not that much of difference. So that is the first SARS-CoV virus analysis. We found to detect which one is the healthy and which is wild type, and then all this. That means the Omicron, Delta, and healthy one, that is no such non-infected, and infected status, and then different versions of that. So that is the molecules, okay, the protein molecules. Then we try to shine but it is the FTIR spectrum, here is 51 and 1225 spectrums. So you can get the first observation with FTIR. But I said that we try to figure out the Raman spectrum of this Wuhan Delta Omicron virus. And then we found that how can I cluster that? Which one is what? So we make roughly 473 samples and train the data with their spectrums in a algorithm, and then we have been able to, you can find from this one, the non-infected one is the red one, and these are the wild type Delta and Omicron versions. Those viruses we can make the clustering of them, and it is very quick. So the standard way, it takes a lot of time by PCR. But here, just within a minutes, we can actually make, using that AI platform, we can clusters and find out that what is the distributions of those different samples, whether it is the Delta or Omicron versions. First is the identifications of the kind of virus, and then we try to predict it, the confusion matrix. So if you look at the confusion matrix for that, we find it very interesting. H for Wuhan virus. So we have predicted 32 Wuhan virus case, and there is no false state. So 32. If you look at the Wuhan, this is the healthy one. This is Wuhan, this is Delta, this is Omicron. Only in the Omicron case, you find that there is a false detection of the Wuhan, but actually it is Omicron. And so there are actually 70-30, 70% trained data and 30% is the test data. And we found that 98.6% prediction is possible by the Raman spectrum and then by the further its cluster. So whether this can be used as a clinical spectroscopy for a marketable products, that is the idea, that it takes nearly 2 years at that time to develop with this Raman. That's why the Raman setup is there. And ICMR then came forward and they want to fund this very interesting work. But one thing we try to solve is how to increase that strength of the signals. Because I said that Raman signal is weak. Then we have used the asymmetric circular resonators, split ring resonators. Meta surface we have created on silicon dioxide, and it is two such things, and with one is one angle and another one is a different angle and the radius. And we actually broadcast the samples, spread it, which is within this ring, outside the ring and take the Raman simultaneously. And we found, you can find that because of the surface plasma resonance, so we are getting an enhanced versions. Signal gets strong. So that is the meta surface we created. It is by UV lithography you can actually make it. And then if you look at the spectrum, this is the normal surface, but with this meta surface, there is a profound enhancement of the signals. So we can get a better, I think that your observation will be much more clear if you enhance its pattern. So that is one thing that ASRR based asymmetric speed beam resonators based spectrum analysis for amino acid is the protein structures, and for Omicron, human, we find it for other viruses too. So now these innovations, or I would say these observations, can lead to a good, very quick analysis for these detections of virus. So both the Raman spectrum and their meta effect taken into account. So we give and it is pretty simple. If you in my lab we just get that mask and make it. It is a gold one, and you can find the difference of its radius and the angle. So, and you can find that it is not so difficult to fabricate, and you can enhance this way. So with this, now I say the next part. With this in the innovation, so I got a good paper in analytical chemistry. But we are not happy with that. Let's try to develop these as a products. And then the ICMR, there is a mission project, medical device diagnostics from ICMR. So they have come forward to fund these projects. So we are expecting a product, a company will come up from that. And my younger colleague is Vasudev. He just joined a few years back. He's working with me. And I also got a gifted postdoc, Pooja Lahiri. She is wife of Vasudev. She is actually the main person working with me in this endeavor. So hope that this particular product, the clinical lab, so clinical spectroscopy is coming in a big way. And we have made some sort of this is the whole approach of the business models. And color mapping and etc. So level-free diagnostics techniques, and where pathological assessments can be done automatically by image processing and clustering. So it's spectroscopy, it's image processing, clustering using AI platform, and then final interpretations. So it's a total combinations of science, technology, and AI. And if you look at the market, that also surveyed, because for these medical, they are very keen in the market. So it is seen here of 9%, and this is the 310 million market at the moment 2019. So it's a growth of 9.1%. So we are thinking that to bring a startup based on this clinical spectroscopy. So what I am saying, the message is very clear, that enjoy your fundamental research. It takes years, but finally you think of a product out of it, which is the need of the day for our country. And you just look at it, people are there to help you out. Fund is not a problem. I will come to my next innovations where I will show how fund is not a problem. Next one is there is a main theme of my work is bridging innovations and commercialization. So you understand the perspective. that this is the innovations, you understand there is a market, and you understand why we need it. And ICMR, like those, they have recognized this work, that means that it has a potential. And definitely you will look at here that you have the technological progress, economic development, social well-being, and etc. You have to just club together. So that is the loop you have to follow. So always you remember that innovations, then there will be a pull from the market and push also from the market. So finally there is a coexistence of the product and innovations. Without the product, innovation will not stand, and without innovations, no product will stand. So that is the, I would say, the mechanism behind it. Come to the next one. There are few products we are working at this moment. I will pick few of them because of the time constraints. For example, drug delivery. This could be a fantastic, huge market potential. Why I am saying that? Maybe gas sensings for early detection of diseases. Intelligent nanobots. Why I am just saying. So, MAP, microarray patch for drug delivery. The second innovation that has already got in. So all of you know that it is a very interesting observation. In 21st century, we continue to rely on an outdated vaccine administration systems dating back more than 200 years. Just think of it. Technology has progressed so much, but still your newborn baby, when they are vaccinated, they are being actually vaccinated by those the needles. And so painful, the parents know how is the traumatic situations. So I was thinking that my group, same, again, discussions came, can I do something? Can we do that something? So that it will be a painless and even parent can administer the vaccine without any help or assistance of the doctor or any specialized staff. And you know that this is not only this traumatic scenario, another one is the cold storage requirements. That is, if you look at Kashmir and all, 70% of our vaccine goes west because of the cycle, that is the cold chain cycle is not properly maintained. And it becomes expensive to reach to the people. And India, like 142 crore people, think of it, so many young generations we have. So from 6 to 13 or newborn in 13 years, how to vaccinate them rapidly going to the remotest part of our country. That was the mission. We try to give solutions for that using our known microfabrication technology. So this is, I quickly go that, this is some kind of a patch, okay, having those dissolvable microneedles. And that is the same picture, you can find this is needle. But this needle is made of drugs. The vaccine is impregnated into the polymer matrix. That polymer is the proprietary one invented by us. And with a simple applicator, I just push it, the patch will be on the skin, and 30 to 60 seconds, vaccine will dissolve in the baby's skin. Baby will not even understand something is going on, because it's painless, even playing mood, you can deliver the vaccine. So we are very excited to approach the, again the ICMR supported these activities. Then the ministry declares that this map will go to the national mission for delivering the vaccine for H2H1 flu and polio, not polio, hepatitis. So these two they have identified. While doing that one, then suddenly I got a call from WHO. So I have been into Delhi and we had a long discussion. I I think that was a fantastic occasions to meet all the from African nations, from others, look at their need, and they say that we need for Africa, we need Ebola virus and measles. Can you do a drug delivery platform or vaccine delivery platform for them? Then it's a brainstorm for 3 days. Then we actually these formulations based on what drug you like to use, what vaccine you like to use. One of the USP of our product is that it's thermally stabilized. So you do not need any cold chain. Even at 40 degrees centigrade, you keep it for 90 to 120 days. The efficacy of this vaccine is not changing. It is nothing but microfabrications and little bit of polymer science, micromolding. That's all. But the formulations. What is the physics? There is always a physics you have to understand first. So first the operations, that it will go to the transdermal vaccination program, and all the vaccine companies, they have decided that transdermal, that is not going to the dermis and going to the peripheral regions, sensory nerve, it is a sensory nerve. So you know that they want transdermal delivery platform so that the vaccine can go inside and diffuses slowly and then goes to the blood, and it works. So that is more efficient than the conventional, that is declared by the pharmaceutical. India is #1 in pharmaceuticals. Serum, then Bharat Biotech, etc. They are very, very big producers of that. So the government actually given us to approach them to make it. So that is 30 seconds to 1 minute. You just push that, it will be dissolved and no data. So don't you think that what I am at the moment innovating, other corner in some world, nobody is working on it. It is fool's paradise. You always remember whatever you are now doing it, anywhere in the world, somebody is also working in the same business. Then we found that there are 2 places in the world this is what. The WHO is an eye-opener for me. One company is from Queen's University. They have developed this map, and they are already tied up with the serum industry. And they are making, because it will be in the market like a drug, because drug will be inside. While licensing, you have to be careful that whether it will be a device or drug, it will be a drug. So drug license here. So you have to go to the actual pharmaceutical industry and convince them, and you put a pilot plant there to make that map. So another company, Micron in US, they are also doing the similar kind of, but all in the trial stage, not going to the market. So WHO announced that 2030, if you look at their charter, the map will be the final vaccine delivery platform to reach the entire community, especially for the age group up to 30. There is a declared policy of WHO. So detailed policy has been given there, the protocol, etc., etc. You know that some European guy told us that if you use your patch, whether I will have that colored patch or not, some reddish things will come or not. Even where to place the patch, here or here, my beauty will be affected. So I like to power that one. So all this story of the geopolitical, religious bias. So that's the entire dynamics you have to follow to look at the possible market model. So WHO is an eye opener for us. They are in touch with us. We used to have meet with them. So this is the India map. So government of India, it is the solutions for that India map. And India is the biggest producer of pharmaceutical products. So one of the companies already tied up with us, but they say keep it secret, because once it will come to the market, that is for they are famous for rabies vaccine. So for that and other products, this will be their platform. Now come to the science. Without science, nothing works. So actually the why protein molecule, this vaccine gets affected, loses its efficacy. Because when it goes to high temperatures, then it denatures and it changes from alpha helical phase to beta phase, and so in particular phase is required for the vaccine. So you have to protect the protein from denaturation. That is the key of the science, the innovations. So what we have done, we have made a polymer. We call it caging. So virus vaccine is there in that cage, impregnated into it. This is a proprietary method. This is our patent. Thermal vibrations is there, but my drug molecule is shielded from it. So the protein molecule doesn't have any kind of changes in the phase. It remains unchanged. So that means the efficacy of that, we have checked it. There is a clinical trial for that. That the normal vaccine and this specially platform, the delivery platform, whether it efficacy is the same. We found it same and if not better. Even commercially, it's much better compared to the conventional one, because cold chain not required, loss of vaccine is not there, I need no assistance and so on. And then suddenly a company has come up. Two of my students, I am mentoring that company, is Innovodite. Believe me or not, the first it was we call cohort. I tell all the students, especially, if you want to start your own venture, if you have innovation and strong things like that, then there is the art of the partnership. Then your Glasgow company, they have offered us to train us how to make these products to a market. So after rigorous 3 months training for that, our students, lot of teaching, etc., how to, and what is the key is entirely different. The scientists observe something, but the market people, their approach is entirely different. So that bridging the gap, so that they will treat it. India has got a fantastic opportunity now for their startup. Then the angel funding, they have, this is called paddock. So they approached us and they want to host this. So that becomes like a, that we call it, there is a investor's window, and that window is open for 2 hours. Who will bid for that will win that. You believe me or not, we have a targeted fund, say around 10 crores. I am not divulging the exact value of my companies. Yes. But they have identified this is the value of your company. Within 2 hours, our company is oversubscribed by 1. Just 2 hours window. The company was traded. My capital, I need 10 crores, I get 11 crores. Next day I got a call from Munjal, that is Hero Honda. Hero Honda is making bike, but they called that suddenly we were interested for you. So they say I want to invest. And already another largest pharmaceutical company, they told us don't say our name because we told that we are like a competitor for that. They have already tied up with us for this particular product for rabies virus and some other. So that is what, if you have this science strong, an innovative mind, and look at the market, you have greatest opportunity. And they say that it will be when my preclinical trial run will be over, they will do everything. Preclinical trial, pharmaceutical companies. Then my company's valuation will be 200 crores. And once it goes to the clinical trials and the sample has started to go, it will be 2000. That is their prediction, not my predictions. That is why they are jumping to invest in this India. So this is one of the product that I say, from innovation to the market and what is required. The same PhD students, they say no, we will not go for, they are fantastic students. They have got a lot of offer from abroad, they say no, we will start our company and make it, and they will do it. So, and this is the physics behind it, so we have a patent, 2 patents now. Next term is, this is the value propositions up to 40 degrees centigrade, 120 days as per the WHO guidelines. No change in the vaccine efficacy. Minimal dependence of the cold chain. I don't need any cold chain. That's my USP. And the company that is the twins spin-off, they have a plastic waste. But in our product there is no waste. Just it is vanished in the skin. So I am not, my carbon footprint is ensured. So that's why Bill Gates Foundations, they approach me that we want to fund it. But my government say, don't go for Bill Gates Foundation. There is some I don't. You probably know what is the political significance of that. So vaccinations is a huge market. Huge markets you have. Millions of such vaccine delivery platforms distributors. So to scale up that one. And that is why I am already talking with the vendors. So vendors, we give the design of the machine so that finally we get the product to the time. That company is say producing thousands of map per day or something. Then India gets millions of such. So different approach. So that is also being, that our honorable education minister at that time given a congratulatory message to us. Finally, our home minister Amit Shah as well as our Prime Minister Narendra Modi giving me a congratulatory message for this innovations. And this is, I was invited to G20 summit, and that was one of the leading innovations in G20 shown. Kiran Majumdar Shah, that lady, even was excited. And we also give a complete platform that is not only solid, hollow microneedles for drug delivery with a micropump and a platform for this another products, because every time you have to find new avatar things. Now coming to a very basic science. Yes.
What is the cost actually?
Yeah. First of all, that since it's academic, for your students, especially know that, don't say what will be the cost of your product. That I learned. Suppose you will not going to be the user, right? The market will be done by say Serum or some company, which I am not naming. If you tell the product cost, where will be their market? I may be doing it with 150 rupees. They will be selling it at 1500. But if the innovators declare that funding, so please do not make this mistake. Don't tell the price of your product or bomb cost. Remain with it. But it's an academic institute. So I believe my students, it costs me less than 60 rupees. Vaccine cost is different. Vaccine, I am not doing it. But the rest of the process, yield is, I can manage that. Studies I have to do it. 60 rupees is good enough. You might get it in the market, maybe 600,000. So that is, that is our patented polymer. That polymer matrix hosts this vaccine. The formulation is made such way that it is thermally shielding that, so that protein is not denatured. So the efficacy of your vaccine remains unchanged. Now coming to the next one, drug delivery is the targeted drug delivery. Can we do that? This is one of the first observations having done in IIT Kharagpur in my lab. I have a collaboration with NIMS, National Institute of Material Science, Japan, and also University of Arizona. That idea came first by Penrose, and his student, Stuart, is my collaborator. Penrose obviously knows his work and is pretty excited. He's 90 plus. So I am excited to share my latest innovations in that. So we developed a nanobot. It's a molecule. Okay? It has got motor. It can move, but in nanoscale. With NIMS collaboration with Japan and Arizona. Then Shannon's that maze problem. We tried to solve. What we did, very quickly I go to the innovation side. Yeah. Look at here. Suppose this is the point A, where I inject that nanobot. And here is the cancerous cell. And I give this maze problem. This is a very intriguing thing. There are a lot of, you remember that maze problem we solved, how to reach that target with the minimum part. So, this molecule does have that intelligence. It actually goes there with minimum possible time and path, describing its own route. And I think this is a very long 12 hours, 15 hours experience. I don't know why this video is not working. It should work. So, in video you can see that if there is no cancer cell here, my nanobot would not go around. It will just go around. If there is a cancer cell, then and then it will go. Otherwise it will not go. Second one, I say that if the pH value difference is the reason for that, I actually simulate the same kind of pH value over there. But nanobot which has not gone inside. No, it's still not going. Whenever only cancer cell, it has got the intelligence. It can go through that maze and attack this cell. Absolutely, it will be amazing to know the video of that, which takes 15 hours of experience. I like to share that. This is the first experimental results of a nanobot intelligence. So we try to say that it has got the intelligence. And one thing I checked with the mobile phone, its interaction with the wireless RF, then I find that it is not working. That nanobot starts to go haywire. So that is one thing. Call this effect as magnetotaxis. So it's a new phenomenon we have developed, and you understand why I have that done. This is what targeted drug. That nanobot would have the required drug, and it will reach to a particular affected cell, and it will annihilate. So that is the targeted delivery from that. How much minutes we have? Almost. So last innovations which has gone to the market. So I am just keeping that one. That is another innovations for micropump. For medically for ICU patients, you know that for critical drug delivery, automated approach for using. But another one, if you just look at this video, yeah. So this is the product. So this is the drug. And you see it is connected with Android. And depending upon the patient data, doctor prescribes the dose and the time. So you will provide that dose and the time. So dose is say 10 units, say insulin. Okay, you give the dose, say 4 units and 35 minutes. Then it starts injecting. You will see here. So this micropump that can actually deliver with an accuracy of 0.1 microliter. So that is a good model for us for caring our patients, especially insulin drug delivery. So microneedles connected with that micropump with Android phone, just click it. So what is my next target? Make it wearable, make it small and small. So that is another one, also supported by ICMR. This is the final prototype. Now I need the challenge from my younger students, how to miniaturize that one to make a wearable device. Here all the components have faced innovations. Main problem is, I say that piezoelectric is not the actuators, because piezoelectric materials is not applicable for bioapplications. It is not biocontractable. And don't make any biomedical device which require more than 10 volts. It's not acceptable. So you need to develop something which is not piezo or QLED based. So we developed a special IPMC, ionic polymer membrane, with some doping, light element doping like lithium, and we have reduced the actuation voltage to 6 volts. So that is, and giving a deflections up to 35 micron. It is good enough for pushing the drug to the human skin. So that is one of the patents, and one of my PhD students just got his degree based on that work. I say that each PhD student at the day of their final defense, with one hand at the end of his defense should show it. Otherwise I will not sign it. So that is another product we are looking for the market. I am avoiding this one because this is something triboelectric effects, how we are doing it for traffic monitoring and load bearing. This is published in Nano Energy. Just I am skipping. That is what, there is a triboelectric sensors, and there is a strip between the highways where the motor car or truck loads will actually go through that. It will see that what is the load and how many. You can count, and simultaneously it will also see the load. If the load is more than that, alarm will come. And that is by triboelectric effects we get. So this is the circuits and all this. Now, this is my last topic. Obviously there are many more. Just give me 2 minutes. And entirely different from the electronics microfabrications, the nanotechnology for construction. What I did so, what is my role in civil? But I find a fantastic role in making green cement. That is the product. We have started a company on that. We named this as G-Build. We have used some graphene based stuff, and which has got a unique functionality. I will show the physics first, then I will go that this is the cement. Okay, basically it is calcium carbonates and some sulfur, CHS. And then we introduced an additive, some graphene derivative, which introduces this kind of aging over this CHS. And now look at the imaging phenomena that comes. Next slide. Without admixtures, if you see that crystals are like that, okay, the cements that goes to the concrete and make the strength. But look at the growth of these crystals. Just by introducing graphene, some derivatives of that, I see huge crowd of them. And that is binding energy of this concrete is much more stronger than this with this admixtures. So what I am getting that it is almost like porous fluid. So my water retention is much higher in this. So 70% higher calcium silicate hydride crystal formations. Just it stimulates this catalyst, this graphene based one. And if you look at this range of this, which is without that additive, but with this additive, look at how the surface breathes. So in the same image, that quite clears us the image is, the surface is quite dense. And so, but what is the market from there? What is the physics behind defined? So how are you going to that? Just look at here. I increased my compressive strength by 40% with less than 20% clinker. Do you know the business from that? The clinker is the basic raw materials, which is primarily calcium carbonate. Think of it, if you reduce the 20% cost, that straightway 20% carbon reduction. Straight. So business is quite clear. We surveyed for Adani 1 million ton capacity. We found that in year they will save 2,000 crores just using this additive. So with the cement bag, we'll give a small additive. You just add it with certain portion, and water 16% less reductions. So we named this product as green cement, and a company has started called UniAim. Be my second startup, another student's body. And now who is my customer, you know? Brigade. Everybody knows in Bangalore, huge construction company, the Brigade. And we are, it has already got NABL accreditation. NABL accredited lab, you have to share all this data, you have to share with them, you check. Concrete slab, we have given, then we tested it. So just nothing but nanotechnology, amazing property of nanotechnology. How it can lead to products which have $1 billion. Okay? So that is also doing well, got funding from BIRA, big grant. Now Brigade is, and wholesale, the world best cement producers, they are also interested in this. They say even if you can increase the strength by 6 to 7%, it is amazing. We are improving, I showed that, nearly 30%. Okay, so that is one thing. It is purely physics. Of course, the application is down to art. But just the idea, I don't have time to go to art. So it's already got the bridge, the Brigade group. That startup is on that one, and then it has got a lot of times of media and everything. I think the noticed it. It has come in the newspaper. Of course, these are the things that I want to see finally the product of the month. I think accolades, recognitions is fine. But in my age, I want at least one product to go to my society. I was a lot for. So now next one is coming to the soil. I am working on developing some automated platforms for soil nutrient measurements, automated NPK sensor. You know, there is no sensor exists which detects NPK samples, that is the nitrogen, phosphorus and phosphorus. There are different sensors and very tedious, expensive processes. But everybody looking for a sensors which can automatically, I can give my last slides. This is the physics of that. Look at, what is this? Not space, Mars, which say our next generation, I think the structure. Sure. Okay? And that is the full of sensors and dispensers. So as per your requirements soil, at that grid, they will disperse that particular, say, urea or fertilizer. So no over fertilizations of this. So that is the people. Develop something, not only moisture, I say most of the work, how to sense the moisture of the soil. Lot of papers. But I don't find paper in say NPK sensors. You look at it. And whatever you will do it, make it facile and less expensive. So that one of my PhD students developed that. She has already submitted her thesis. I don't know whether some company will come out of this or not. But still, that approach is how to detect the NPK and isolations of that. Definitely. Soil is a very complex mixture and cross-contaminations and there huge problem of AI and material science and electronics. and the robot will go, it will dig it, take the samples from 6 to 12 inch from the surface, and then it will mix with something and then do the measurements and feedback. It will have a controller that will control and dispense your nutrients. So that is the whole idea. And my one MS students did the earlier work about the design of the truck and the machines. Now the sensors part will be students. So there is more or less another is battery, which I am not. Most of my publications are here, but at the end of my talk, everything what I told you is because of that. Okay? And one thing I will say that none of them are IIT projects. I do not take IIT projects. They are above the sky. I limit them, pamper them. They are all our University of Kolkata, WBA, or VIT Misra. My student is their faculty at the moment. She was earlier a student of here. So they are from there. They are doing excellent work in this one. So this is my team and my lab. And all the equipments, more equipments are coming. The spectroscopy, lithography, and probe stations for making resistance, sheet resistance measure, then high spectral imaging microscope. Those 3 machines in this financial year is coming. Already it has arrived. And finally, I would say my students, in the science day, the aim at heaven and you will get art donations. Aim at art, you will get art. So please, do that hard. Make your ambitions pretty high. Why not you one day will be a billionaire? Okay? So thank you. So those are interested to look at my publications, you can scan it. And this is my lab. I have a virtual tour of my lab, all these setups, which is done by Doordarshan and Charity Pakistan. So that is also in YouTube. You can tell my name and microelectronics, then Doordarshan 1 hour documentary based on this work is done by Doordarshan. So that is, you can find this. Okay? With these few words, let me thank first all my teachers who has made me here. So without them, I will be nowhere. Okay? I am currently the institute chair professor. So I believe that if you have strong will, you can be everything. Even you can do much better than so-called elite institute students. Okay? So I am looking for students only from those institutes. They have the gym to work, show their strength, come to me and work with me. Out of box problem will come to you and it will be solved. If you solve it yourself, you will do it. Okay? Thank you. Thank you for that.